Okay. Yeah, so, well, welcome back everyone to SPAAMTisch seminar. I see today we are many. I'm very excited. So, today we have John Tay, who is a doctoral student at the University of Melbourne and is a visiting student now at Institut Pasteur, hopefully officially soon. And he specialized in phylogenetics and computational biology, focus on molecular clocks and how viruses evolve. He is currently studying the evolution of dengue virus and using advanced models. And in his past works, he worked also on SARS-CoV-2 variants, detecting burst of evolution, improving methods, and analyze virus timeline. So thank you so much, John, for accepting the invitation. And well, the floor is yours, or the screen is yours.
Well, thank you very much, Maria, for the introduction, and also thanks to all of you for inviting me to speak to you guys. So, as Maria said, I'll be talking about Bayesian phylogenetic methods, especially for microbial pathogens, and I know all of you are very interested in ancient DNA. So, hopefully, you can get some value out of this. Um, and as Maria said, I'm currently in Paris, but I'm still a Melbourne University student, but I've gotten a scholarship funding for transferring to Institut Pasteur, so I'll be starting here very soon. Happy to be here in Paris. So without further ado, okay. Um, as mentioned, this is just some of my research projects so far and what I want to do going forward. My first project was on the driver of the emergence of SARS-CoV-2 variant of concern. So I'll be talking about that. I'll also be talking about branch-specific rate acceleration and how much statistical power we have to detect these events. And also more of a theoretical question, but how reliable is providing sampling time information for calibrating the molecular clock? And I'll be going through what the molecular clock actually means. And finally, for work I want to do in the future on dengue virus is the question of how we resolve heterotachy. And heterotachy is when there is branch-specific rate heterogeneity, so rate differences between different lineages. And so I think the best way to start off is just explaining what the Bayesian phylogenetic model actually is. Some of you may have seen this diagram or variants of it, and it's from Duplessis and Stadler, and I think it's a very good overview of what actually goes into the Bayesian model. And essentially, all of these. Can you see my cursor? If not, I need to get the pointer out.
Yes, we see it.
Oh, you see it. So you can see me moving around. Okay, cool. So all of these are parts of the model, and this is the data. The data being the alignment, and the alignment is generated from your sequences. And this component in blue here is known as the phylogenetic likelihood or the tree likelihood. And if you know anything about the tree pruning algorithm or Felgenstein, then you know that this is exactly that. So no different from a maximum likelihood method. The rest of the model includes the priors. So the priors on all of the models that you include, and I'll go through them. So you have the clock model, which is of particular interest to me and my supervisor, Sebastian. Substitution models, which model evolutionary processes. The branching model, which is just priors on the tree and the population. And finally, the topology, including the branching, the actual branch lengths, which come from the other components of the model. And so that's why the tree is dependent on the branching model itself. And so it looks something like this. You have what we call the posterior or the joint, and I'll explain why there's a difference, which is the probability of the model, so all the components of the model given the data, and the data being your DNA sequences, and the phylogenetic likelihood, which is the data given the model, multiplied by all the priors on the model, and you divide it by the probability of the data. But because we use something known as MCMC or Markov chain Monte Carlo, which the common implementation is Metropolis Hastings, which don't worry, you guys don't need to know unless you're interested. You don't actually need to calculate this bottom part. And it involves many integrations, so like 5 integrals on integrals. So it's basically impossible to actually have an analytical solution. So there's no closed form solution for this. But it is actually somewhat important, and I'll mention why later on. This part that you see here with the time tree and the branching model, sometimes people call the phylogenetic likelihood, and that's quite important for phylogenetics, which is not what I'm talking about today. But essentially, if you're interested in epidemiological parameters like the R0 and estimates like that, then this is quite important. But for our sake, we only use this to get the trees, the topologies, and the branch rates that we're interested in. And like I was saying just now, the probability of the data, so the probability of the sequences, is known as the marginal likelihood, but we don't need that right now. But I'll explain later. So I'll be stepping through each component of the model and then start talking about how I actually use these models. And it's sort of a tutorial on Beast, but if we have time, we can go over Beast if people are interested. So the first thing that you can do, or the first thing that you have to do, fossils is spelled wrong, is enforcing monophyly. And you can do that in Beast by defining sets of taxons. So you can say that taxons A, B, and C should be monophyletic. And when Beast draws the tree, generates the tree, then you'll have a clade. And you can also use it to enforce known node ages. And this is important because rates and times are unidentifiable. And what do I mean by that? Basically, if you have divergence, so if you actually know the amount of changes per site, but you don't know the rate and you don't know the years, then you can neither get the rate or the years. But if you have the divergence and the rates, then you can get the times. If you have the divergence and the times, then you can get the rate. So with sampling times, which is what we usually use in epidemiological context, you can provide calibrations for tips. So you can say that my tips have a certain date. And with more ancient data sets, you have dated fossils where you can provide node calibrations. So place a specific time on certain nodes in the tree. And it'll end up looking something like an ultrametric tree or a non-ultrametric tree. And the difference is ultrametric means that all of the tips are contemporaneous, so they all have the same time and it's all in the present. Whereas non-ultrametric, so if you did provide sampling times for, let's say, all of these tips, then you would have a tree that looks like this. So not all the times, not all the tips fall at the same time. And again, this is not a calculation that you need to do or I need to do, but this is what all of the tree generating methods do. And essentially, you can estimate the clock rate if you have the distance in time between species A and B with an outgroup. And you also have things like the time to the most common recent ancestor between all three. And here's where you plug in the fossil calibration, for example, if you wanted to figure out the time to the most common recent ancestor, most recent common ancestor. And moving forward, we also need substitution models. And these are models of sequence evolution. You don't really need to know the math behind this unless you plan on doing more like studies on site-specific patterns. But the key components are this instantaneous rate matrix, which I'll explain in a bit. But essentially, if you look at all your nucleotides, A, C, G, and T, you can get a rate between each of these. So a substitution rate between A and C, G and T, and so on. And the most basic of these models is the JC, Jukes and Cantor, which enforces that all of the rates are actually the same. So there's no free parameters at all, and you're just treating every substitution identically. You're also saying that for all of my nucleotide frequencies, so of A, C, G, and T, they're all 25%. So no matter what the data look like, they all have equal probabilities. And that's what the JC model enforces. This is what the instantaneous rate matrix actually looks like. Again, you don't need to know, but you do like row reductions and you'll get certain values. and you can use, for example, Felsenstein's tree pruning algorithm, and you compute site likelihoods using this matrix along with your clock rate and your nucleotide frequencies to get the actual likelihood of a particular site. And then with each of these sites, you can cover the likelihood for the entire tree. And a few other models that you might have seen that people commonly use, even for your ancient DNA, are the HKY. So the difference between the JC and the HKY is that it accounts for transitions and transversions. So between purines and pyrimidines, the rate is different. And you will get something called a kappa parameter. So this kappa parameter will be estimated by Beast. And it will determine the difference between transitions and transversions. And finally, another commonly used model is the GTR, or General Time Reversible Model. You call it like the most advanced substitution model, but advanced in the sense that it's still using what we call the CTMC framework, or the Continuous Time Markov Chain framework. Again, don't need to know that. But if you're interested, it's always good to learn these things. And the main difference is each of the rates between each of the nucleotides can be different. So A to C can be different from C to G, and so forth. However, it's not that it's always different. It's just that you then estimate these parameters, and sometimes you may find that the A to C rate is actually the same as the G to T rate. It really depends on the data set. But it allows for a more flexible model of nucleotide evolution. And so you get these alpha, beta, gamma, and so on, up to zeta parameters. And of course, for both of these models, you can estimate the nucleotide frequencies. And what that means is, if you choose to estimate nucleotide frequencies, you're saying that at each particular state, I'm going to make my best estimate on what I think the proportion of A's there are, T's, G's, and C's. You can also fix them empirically. So you can look at your data set and say that 30% of my data set contains G's. So then I'm going to fix it at 0.3. And same for C's, T's, and A's. Another important model we use is known as the among site rate variation model, which is also known as the rate heterogeneity among sites model, which is also known as the discrete gamma model or discrete gamma distribution. So all of these things mean the same thing. And what that essentially does is you can have discrete rate categories, so like blocks, and apply these on sites across your genome. So the most common is known as like +4, gamma +4, or when you have 4 gamma categories. And essentially, you're breaking up your gamma distribution, which if you have alpha and beta values of 0.5, it'll look like this. So a very left skewed distribution. And then you would assign a 25% probability on all of your sites. So you're saying that at this particular site, there's a 25% probability of my rates falling here. And if they fall here, then you're saying that the rate is very low. So a very low boundary. And then another 25% of my sites will fall here. So the rate is still quite low, but it's actually higher than if they fell here. And then the rate increases and so on. So you'll have a quarter of sites evolving very slowly, less slowly, kind of fast, and much faster. And that's how you have site rate heterogeneity. And of course, you can also have invariant sites. And that all that is saying is that I'm going to have a proportion of sites in my alignments that don't vary at all. So it could be like 0.1%. And then it means that these sites don't change in the model. And if anyone is confused at any time, like please do interrupt me and ask questions, because a lot of this is very like theoretical. So moving on, we have the tree branching model, or tree prior, or the branching model. And today I won't be talking about birth-death models. They're complicated, and oftentimes you don't really need to use them unless you are interested in epidemiological parameters or phylogenetics. So we use the coalescent process. And what the coalescent process basically is that if you have 2 tips, you will go back in time until you find their most recent common ancestor, and then you'll have a node. So then you have the child and the parents, or the 2 children and the parent, and it's a backwards in time process. And the most important parameter we actually borrow from population genetics, which is the effective population size. And that is just a value that's proportional to genetic diversity, which sort of represents the amount of chromosomes floating around in individuals which are breeding. And there are two main models that we use for tree priors, which are the constant size population or constant population size and the exponential growth. And for this particular, what we're interested in is actually a negative growth rate. You can actually parameterize it with either a positive or a negative, and that will just affect whether a growth rate less than 0 is an expanding population, or whether a growth rate greater than 0 is a shrinking population. So this value was positive instead of negative, then we just flip these symbols here. And what does that actually mean? So if you think about it, we start at n0, the initial population, and we expect something small. And obviously with exponential growth, your population increases exponentially over time. And then it will be reflected in a tree like this. So essentially, as we get closer to the root, our branches will be shorter. As we get closer to the tips, our branches will be longer. And with a constant size population, you will have something that looks like this, which is a bit more uniform. So the distance between each of the nodes is kind of uniform. And I can clarify more later on if anyone wants. And these are in one of the Beast 2 tutorials. And there's a lot of literature on this and which models to use. And so we come to my favorite topic, and also Sebastian's favorite topic, in fact, which is the molecular clock. And it's based on this hypothesis, or the molecular clock hypothesis, which suggests that there is a constant rate of substitution as a measure of the rate of evolution between your taxa or your samples. And so the simplest model that we can define is that there is a single global rate of substitutions across all branches in your phylogeny. And you're able to visualize this in something, a function known as the root-to-tip regression. And all that is you plot the genetic divergence as a function over time. So you have all of your samples here, and you draw a line of best fit through your samples, let's say SARS-CoV-2. And then the slope of the line of best fit you'll get is the rate of substitution in terms of substitution per site across some time. And the x-intercept that you get is the root height of the phylogeny, which is also it's like equivalent to the root age. However, as you can see here, do you think that this is a good root-to-tip regression? Like, if anyone has anything to say about this, like, do you think that this looks like a good regression? Does anyone think it's not a good regression?
Yeah, I don't think it's a good one.
Yeah, exactly. It's a terrible regression, right? Because, okay, there's something funky happening here, right? Because a good regression that we expect is everything like falls on the line. But you can see these points here and these points here and these points here. None of these fall on the line. So I'll be explaining why later on, but we'll come to that. And this, do you think this looks like a good regression? And if you do, just tell, like, just give me some reasoning and anything, like, there's no wrong answers at all. But is it a bad regression? Is that what I'm trying to suggest? Okay, I'll assume everyone's too scared to answer. So no, it's not a good regression. Why? Because if you think about it, like I said, the slope is the rate of evolution, rate of substitution. There's no way in hell that the rate of substitution could be negative. It doesn't make any sense. Like, you can't have negative substitutions, as time goes on. So no, so this is bad. There's also another issue with this data set, which is date rounding. So, well, I gave it away, but why are all the points scattered out like this, like, on a single X line vertically? It's because they're date rounded, which depending on the data set could be a huge issue, especially for epidemiological studies, because you lose a lot of information. Ideally we have the exact sampling time up to the day, and you're able to get better estimates. But because we don't here, and there's something happening with this data set, that we're getting a very bad regression, and that means that we're getting a bad signal, and what we would call temporal signal. Like some of you may have heard of phylogenetic signal, and when we're considering sampling times, we call it temporal signal. But I'll get back to these at some point. So we have this clock hypothesis, but we can actually have more complex models of the clock. And one of these families of models is known as local clocks. And these allow you to define branch categories that can possess different rate parameters, so different branch rates. And they're known as fixed local clocks, or a subset of them, because you can fix certain branches and assign certain branches to categories. And what do I mean by this? So in the study that I did with SARS-CoV-2 variants of concern, you can see here that we have toy phylogeny with different clades of variants of concern, alpha, beta, gamma, and delta. And what we can do is actually define a background and a stem branch category. So what I mean by this is, if you just look here, there's a single rate. So this is a strict clock, single rate on all the branches. But we can have a situation here where the branch leading from the crown, so leading from the clade of the variant of concern up to the parent node, stem, we define as a stem rate. So all this branch is stem. And we apply that on all of these stems. So the stems leading up to clades of variants of concern. And you might note that it doesn't include the variants themselves. So we set it so that these branches have a certain rate, and everything else, which we call background, has a certain rate. You can also tell that this is a nested model, which is kind of important, but I'll explain later. And we didn't actually just test these two models. We tested a lot more. But again, this isn't, you don't have to remember any of this. But we tested models such as having the 4 different branch rate categories. So having different rate for the stems leading to variants of concern, the variants themselves having a different rate, and so on. And we tested all of these. And the one difference between all of these or the one thing to note is this relaxed uncorrelated model, which is a bit different from these fixed local clocks. So with the fixed local clocks, I define these categories myself. With a relaxed uncorrelated clock, what I'm doing is I have this underlying distribution. So it could be like a gamma distribution or a log normal distribution. And what I'm doing is for each of these branches here, I'm just going to pull a random value from my gamma or log normal distribution, and then I'm going to assign it to the branch in the tree. And so all the branches end up having a different rate. There's a family known as autocorrelated relaxed clocks. So autocorrelated, that means it's like kind of correlates with itself. That basically draws rates from the branch that's the parent, and then you will end up having branches that are related to the parents and so on. But that model doesn't perform as well. So typically, for most studies, including ancient DNA studies, people use the uncorrelated clock. And you can kind of tell why that's better, because it's able to capture more rate variation. If you just use this model, you can't really capture any rate variation. I mean, you can't capture rate variation. But if you have some sort of biological intuition about rates on certain lineages, then you can have a fixed local clock, for example. So back to SARS-CoV-2 specifically, by using the fixed local clock with shared stems, so having stems have a single rate different from the background, we found a 4-fold increase in the rates of these stems. And this is just a violin plot, which is a posterior distribution flipped on the side, and it's symmetrical. So if you've ever opened a beast log output and put in a tracer and clicked on marginal density, then this is exactly what you see, except it's rotated and it's symmetrical. So this size is the same as this. And if you take the mean, so like roughly here and here, it's like pretty much over 4 times increase. And that's basically representing what we call episodic evolution, which are short mutational bursts where there's a large accumulation of substitutions in a short period of time. And I guess you could call them hypermutator lineages. as a shout out to Maria's recent paper in Science. And if you look at root-to regression, as I was showing earlier with the crappy example, the lineages with a lot of mutations will end up as a cloud of points above what you would normally expect to see. So for dengue virus, that's exactly what happened. And I think my next slide, no, after this. Okay. Yeah, so what can we actually do with these models? So like we've run these models, but how do we actually compare them? Because like you'll get trees for both, you'll get parameter estimates for both, but it doesn't tell you whether one model is better than another. And some things you could do include Akaiki's information criterion, Bayesian information criterion, likelihood ratio test. There's a lot of different tests to test models. However, everything or all of these tests have different limitations. So with the likelihood ratio test, you need nested models. And nested just means that one model can be transformed into another by fixing certain parameters or are reducible from one to another. And for AIC and BIC, the issue is you get scores at the end, but the scores don't tell you like the magnitude of, is this model twice as good or 3 times as good as this other model? It doesn't tell you that. You just get a final score. So we arrive on Bayesian model selection. And it's that probability of the data that I was talking about earlier, the thing that we normally don't calculate. It actually turns out it's quite useful, the marginal likelihood. And you can estimate it. So there are ways of cheating to get not the real value, but estimates of the value in methods known as path sampling, stepping stone, and generalized stepping stone. And generalized stepping stone is implemented in Beast. So that's what I would use right now. But I'm sure someone smart will come up with something better soon. And we can use these to get Bayes factors. So Bayes factors can compare pairwise log marginal likelihoods. Okay, what does that mean? So the marginal likelihood is what we estimate using generalized stepping stone. It's in a log value because these probabilities are so small that you have to log them, or you just get numerical instability and the computer goes crazy and can't calculate values. So that's why they're in log units. And then what we can do is assess the support of any model over an alternative model, even non-nested models. And they could have any number of parameters. So you could have a model with 50 parameters and a model with 100 parameters. If you get the marginal likelihoods, you can compare both of them. And so in the SARS-CoV-2 study, we just took the marginal likelihoods of each of the models, and you can divide it by the marginal likelihood of the actually it's minus because it's in log units. So keep that in note. If it's normal Bayes factors, it's a division. If it's log Bayes factors, it's just a subtraction, because you can, when you multiply these without log, when you log transform something, you can just add and subtract, or you should, not you can. Yeah, so we have these log Bayes factors. And what does that actually mean? So if you look at the models that we tested, and you look at the fixed local clock with shared stems, that performed the best. So that's, of course, set to 0 in terms of relative terms. And these are negative because they're worse. So if you compare the strict clock to the stems model, it's 13 times worse, and so on. So actually, the uncorrelated models perform quite well, and so does the local clock with stems having different rates. So here it's like 4 stem rates plus the background. Here it's just 1 stem rate plus the background. And then this is the gamma uncorrelated. So this is drawn from a gamma distribution when we assign the branch rates. And this is just drawn from a log normal. And the rest just perform a lot worse. So I'm not even going to talk about them. And again, if you have any questions, please ask at the end. Now, this gets into a more theoretical framework about how we test what we call sensitivity and specificity, which is like, your type 1 and type 2 error rates. I won't go into super big detail, but I'll try and spend enough time on this for people to understand why this is important. So I'll start from the beginning. What we did was we took our SARS-CoV-2 data set, which I should have mentioned was 200 samples from the 4 variants of concern lineages, and also from other SARS-CoV-2 sequences that weren't very of concern. So what we did was that we took that data set, then we simulated, so we generated 100 phylogenies and 100 sequences that were based on the true data set, but now have these aren't actually real trees or real sequences. Then what we did was we applied a rate multiplier on the stem branch. So we literally just like if you can chuck the tree into, I don't know, R, and then you just multiply the stem branch by some factor. So here it's like a factor of 1.5 times, 2 times, 3 times, 5 times, and 10 times, so on. So then this is like a simulated rate acceleration. And then you can test these with the log-based factors, marginal likelihoods that I talked about just now, and you'll get different levels of support. So if you look back on the Cass and Raftery, very important paper for us Bayesians from 1985, if you have a log-based factor of 0, then there is no support. If it's greater than 1, you have positive support. And only when it's 3 or greater can you start saying that there's actually quite strong support for one model over another. And 5 is very strong. So 5 is usually conclusive evidence, I would say. So here, what's going on here? All of these points are, each of these points is from the 100 phylogenies that we simulated. And each of them, we compare between the strict clock and the fixed local clock model that we tested. And you can see as you increase the simulated rate acceleration, the level of support increases. So it's very good. Like if you look at just a non-accelerated data set, we almost have, we have 0 detection or zero very strong support simulations, meaning that the type 1 error rate is 0 when there is actually no signal, which is great. And as you increase the level rate acceleration, you get more and more support, to be expected. So what we found was like a 4 times increase. And if you look at these simulations, that's quite a good, this is basically quite good evidence showing that the method's robust. Because if you see a 5 times increase, then you can be very confident that it is a real 5 times increase. But that just means that, if you have data sets where the increase is only 1.5 times or 2 times, you might not actually be able to detect whether there has been a rate increase or not. So the sensitivity is strong, but the specificity, so type 2 error rate is a bit higher than the type 1 error rate. And I hope that wasn't too confusing. Now, back to the really crappy regression I showed earlier. If you chuck it into Clocker2, which was created by my former colleague, Leo Featherstone, very good tool. It's like Tempest, but like it's online and everything runs online, and yeah, it works most of the time. Well, tell him that I said it works all the time. You can actually select a number of clocks to apply on the tree. So if I select 4 clocks, then if you look at this, yay! If you know much about dengue virus, you might recognize that each of these are dengue serotype, dengue 1, 2, 3, 4. I don't know which is which here, but this looks way better because if we run like 4 different regressions, and then you get really good lines of best fit, or much better than a single line going through all of them. So what's going on here is, yes, there's definitely more than one rate of substitution, more than 1 clock going on here, possibly 4 clocks, possibly more. So, we don't just run regression and call it a day, we have to look more into what's going on. But again, I'll talk more about that later. So we come to a very popular tool, especially for phylogenomics, and testing whether enough time has passed for there to be temporal signal. And essentially, what the test does, known as a Bayesian evaluation of temporal signal, is how do we actually test for ultrametricity? So we're testing whether by providing sampling times, whether that improves the fit or whether it doesn't. And sorry, I'm beating around the bush, but it's whether the calibrations capture enough genetic information. So As I said earlier, ultrametric trees look like this. It means that all the samples are contemporaneous, so all of the samples have the same time, usually present time, and non-ultrametric when you provide sampling times, and they have different times. And so this is sort of the issue that we were seeing earlier with Powassan virus. It's a mixture of date rounding, but also the fact that usually when you provide sampling times, your clock gets calibrated, right? So when you provide sampling times, you expect a line, but by providing sampling times here, we have not increased the clarity whatsoever. And there are a lot of reasons why this can happen. I'll go through one of them, and I don't think it sort of does apply for Powassan virus. And the phenomenon is known as tree extension. And you'll see it very soon as to why it's called tree extension. Actually, I'll just show it now. So you have, these are simulated data sets from Powassan virus. If you just analyze them and run your beast model, you have clocks that look, trees that look like this without sampling times. So the true model here is ultrametric. So we're saying that in reality, all of our sampling times fall on present time, they're contemporaneous, and so you get a tree that looks like this. Now, what happens when you put sampling times into Beast? When you add the sampling times, it completely deforms the tree. So then you go from a tree that looks like this, then you add sampling times, and suddenly your root age went from like 2 to 1,000, which is terrible because that's like a 500 times increase. So you've completely shifted your time scale. And essentially, this is a form of type 1 error. And that is when you detect temporal signal when it isn't actually present. So you're saying that there is support for uninformative sampling times. And that's reflected as a better model fit for the incorrect model, which is the model with sampling times. So yeah, you provide these uninformative sampling times, and you get an overestimation in the tree height. And really, these sampling times don't provide any calibration of the clock whatsoever, as seen here. And so why does this happen? Honestly, we can only make a few guesses as to what happens. We're not 100% certain, but it's very likely that the model is compensating for the likelihood penalty imposed by including these incorrect sampling times. So what do I mean when I say that? So normally, when you don't provide the sampling times, you'll get a root height estimate, and you'll also get a phylogenetic likelihood. Now, what happens when you include these sampling times? Well, the MCMC still wants to sample the entire space, and of course, it will try and converge on some likelihood, and you end up getting a very similar likelihood, but to compensate for the way that it compensates to reach this likelihood is by increasing the tree height. So you'll just get these trees that are overextended. And what we can do to fix this is place a uniform prior on the tree height. So these are known as bounds, and they're strict bounds. So it means that if we place a uniform from 0 to 5, 5 is the hard cutoff. So most of the time we see that it will, all of the entire estimate will reach 5 because it really wants to be 1,000, but because we've placed a hard bound, it will only reach 5. But at least you end up with trees that look like your original trees within some level of same order of magnitude. And you'll end up also having a lower likelihood. So it makes sense. This is why this is the primary explanation that it's a likelihood compensation, because when you place these bounds, you get a lower likelihood, which is not a bad thing because essentially otherwise we'd be saying this is the truth when it's clearly not the truth, because we know our data, and our data is ultrametric trees that should look like this. So I hope that hasn't been too confusing, and I hope that is a good example of why including sampling times doesn't always make sense, or you don't always get the results that you want to see when you include sampling times. And finally, I'd just like to talk about my future research directions, which is when I moved to Paris, essentially. And it is specifically the question of if there are particular mutations or sites that coincide with spillover and spillback events in dengue virus lineages, and also what the role of episodic evolution is in the long-term natural history of dengue virus. And the way that I'm going to address these questions are Markov modulator models, which are a subset of mixture models. So I told you about like the GTR and the HKY, and I told you that they're CTMCs, continuous time Markov chains, and these just break those assumptions. So you no longer have a continuous time Markov chain. And by breaking these assumptions, you can have a more flexible model. So you will see time and time again that the more strict your model is, the more assumptions you make, the easier it is to calculate, but then the less realistic it is, the less realistic model of evolution it is. So by relaxing more and more parameters, we have more complex models, but that also means they're harder to calculate. But at least now they can be branch and site specific. And what I mean by that is, for example, if you have this dummy alignment here, and then you have a T here and a C here, I call it alignment, but I mean, it's not aligned because there's only one sequence. But you can apply, for example, the Q matrix, so the rate matrix on this particular site, and also have a different rate matrix on this other site, but also have a switching parameter. So you can make these switch between Q1 and Q2 across different times. So it's a very, very flexible model. And these are also reflected in the trees. So that's why it's both branch and site specific. And you're also able to pinpoint mutations responsible, or we hope we can pinpoint particular mutations responsible for species jumping using these family of models. And yeah, that's my talk. I hope it hasn't been too confusing or rushed. And I have to thank everyone I've worked with at the Doherty and the Pasteur, and some of my collaborators who worked with me on my projects up to now, and everyone in my current team in Pasteur. And thank you all for listening and giving me your attention.
Thank
Yep.
you. Yeah, Irina.
Hi, thanks for this talk. It was very helpful because I'm trying to start to get into dating with samples that I have from using calculus. And
Yep.
I have a question about the times that we use, because with archaeological samples, we basically don't have definitive dates. We have a range that's either based on archaeological dating or a midpoint in radiocarbon dating. So
Yep.
do you have a sense of how uninformative that kind of dating is, or whether it can be reliable?
So okay, as always, it's data set dependent, right? So it depends on, first of all, how many modern sequences you have. It depends on how many of these, your ancient samples as well, and whether you have ancient samples. Because if you look at some papers from Stadler and Rachel Warnock, or Tanya Stadler and Rachel Warnock, they do the fossilized birth-death process. And I think that's quite a good model if you're looking at using fossil sampling times. So you don't need, obviously, with the fossil, like you're not going to have a DNA sequence for a fossil, of course. But yeah, it depends on the data set, depends on your time scale, it depends on the rates, depends on how clean your sequences are. Like there's so much that you have to consider, and I'm no expert in any of that right now. Maybe in 50 years.
Do you have a
Yeah, sorry, go on.
Do you also have an idea of whether the rates may be tied to physiological properties? I'm thinking of bacteria here, not viruses, of whether they are slow growth, like anaerobic or spore formers or things like that. Are there publications that discuss how those characteristics might be taken into account when you're doing model selection?
There definitely are. Okay. So of course, I'm going to be always speaking from like my experience, and for SARS-CoV-2, it was the two main hypotheses were that one, that you had patients with chronic illness who were immunocompromised, and then if these patients caught COVID, then they wouldn't their host immune response would be much weaker, and therefore the virus accumulated a lot quicker, and a lot more mutations would occur in these individuals. So that's one hypothesis. And I guess there have been hospital testing, and if you look at these patients, they tend to have more of these mutations. And the other hypothesis was that there are specific like host jumps that occurred, which then led to specific patterns of mutations, and these could also, cause hypermutations to occur, or episodic evolution. I'm not sure, like it's very hard to answer the host jump things because, or the host jump hypothesis, because in my opinion, we're not sampling. That's Sebastian, my supervisor, if anyone's wondering. So yeah, with these host jumps, I think oftentimes the issue is we don't have samples from intermediate hosts, right? We don't have all these populations sampled, so we can't really make a statement on whether that's caused these accumulation mutations. And I don't know what the answer is for that. But in your particular case, I can't say for sure that you'll find literature saying that anaerobic evolves faster or slower or anything like that. But it will be there. Like if you search thoroughly enough, you just need to know the right terms. So like look for rate papers with your organism of interest, and like either a substitution rate or evolutionary rate. Usually that works.
So a lot of the organisms that I work with are species that are uncultured. Do you have any advice for trying to do molecular dating with those where you're definitely not going to have that information?
Sorry, did you say
They're uncultured.
uncultured?
Yeah, uncultured. So we have no estimation of how quickly they grow in individuals or in culture because we just can't grow them.
Hmm. Yeah, I'm not too sure then. What you can do is, so I suppose you have checked the literature, and you haven't found any information on like the potential rate.
Yeah, no.
No, okay.
No.
That's a bit tougher because like for us, we do most of the microbes that we look at are well studied. So like if you look up the rate, you can sort of find the rate, and then when you estimate your rate, you can be like, yeah, this is within some order of magnitude, like 2 times faster. So it's still okay. So it's much tougher when you don't have the rate. Um, but well, if Maria has a good answer, I hope she can help. No, okay. No, sorry. Yeah.
I don't have an answer. I was wondering whether you can let the data talk in that
Yeah,
case.
exactly. You can let the data talk. It's just that you won't have a bench, you won't have a point of comparison. Like you can run it, you can get a rate. Of course, you won't be able to compare that rate to any existing literature if there isn't any. So, it's a bit of a question mark, but you can again you can test different rate models and test different models in general, and maybe look at like related species and see what the rate is in them. Like, there's different things that you can do when you don't have a pre-existing literature on your exact organisms.
Thanks.
No worries. Mina, if I'm saying that correctly.
Yes, hello. Thank you for your talk. I have actually a couple of questions.
Yep.
And first one, I was wondering, what do you take into your alignment? Do you take the whole genome or some of the genes?
Yeah, so well, in terms of like, because I work mainly with single-stranded RNA viruses. So far, not saying that I wouldn't work with other microbes, but because it's like the genome is usually really short. So of course you would want to take as much information as you can. So you take whole genome if you ever can do that, and the genome isn't too large. That's what I would do. But something like I forgot. For example, if you're Beast doesn't, or Bayesian methods, but Beast, for example, doesn't like huge sequences with a lot of taxa. So if you have 200 base pairs and like 1,000 taxa, like your run will never finish. It won't. I mean, it will finish, but like it will take forever to converge. So that's generally not recommended. Something like 2 to 500 taxa or samples with 10 to 20,000 base pairs. That's reasonable. But again, this is all dependent on the data set, right? if your data is like mostly clonal and everything looks kind of the same, then it'll run quicker. But again, it depends. So if you have, let's say, a human genome, of course you wouldn't run Beast on the full genome. You would go gene by gene, and you have to account for things like or think about things like recombination and horizontal gene transfer. So Beast normally doesn't take into account plasmids, but there are certain models that I think Sebastian has developed that can take into account plasmid transfer. So yeah, there's always like there's a lot of things that these underlying models don't actually process. Like even something like if you have a haplotype group and like you have like linkage between certain SNPs and then they all get carried together. Beast doesn't care about that, right? It will calculate the likelihood as if that doesn't happen. And same with recombination and so on.
Mm. Yeah, thanks. I was wondering that because I'm studying bacteria and I was making a core gene alignment of that specific species that I was interested in, and I was thinking if that works or if it's too much, but I was able to finish it. But yeah, and the other thing was that what if like because I didn't have that many reference genomes. I had 16 in total, and only 6 of them had collection date. So, what would you do in that case? Would you guess the collection date for the rest of them to put approximate?
Yeah, so what you can do is estimate the times for each of these undated sequences. And then so Beast would end up estimating a posterior distribution of the sampling times. So then you might have let's say 1995 to 2000 for sequence A, and then for sequence B, 1996 to 1997. So it really depends. Again, like it is a question of like, do you want to let the data speak? You can also place priors, as I said. So if you have some idea, if you know that it's like a 20-year age, like potentially within that, you can set a prior on the age of that particular sample and so on. So there's different things that you can do to tackle questions of uncertainty. I would certainly keep the sequences if possible, because like the more sequences you throw out, the less data you have, and then if you don't have enough data, then your results you won't be able to interpret the results well. Like it may not converge, or there could be other issues. So typically, the more data you have, the better it is, even if they don't have sampling times.
Yeah. All right. Thanks.
All right. Any other questions? If anyone was, oh, Kelly.
Hey, great. That was so great and helpful. Thank you. I have a question also related to what goes into your alignments. And I was wondering if you had feelings about whether or not using just variant alignments biases things. How does that bias things? Should we be just concatenating genes?
Okay, yeah, I think I get what you're trying to say, because like some people just do like just get the SNPs.
SNP alignments, yeah.
Exactly. So the first thing is, if you do just SNP alignments, then you have to remember to rescale the rate. And what I mean by that is, if your genome is 10,000 base pairs, and your SNP alignment is 1,000, whatever rate you calculate is actually just 10% of what it should be, or it's 10% higher than what it should be, right? Or 10 times faster, because if your 9,000 nucleotides are completely completely identical, and they haven't changed at all, then what the rate that you're estimating is actually not reflecting the full genome rate. So it's a question of, are you looking at the mean rates of the organism? Are you looking at gene specific rates? And like it's not a problem to look at either of them in isolation, but of course you should be looking at specific genes while also looking at the entire genome. In terms of just running SNP alignments, I think so far it depends. Again, it depends on your organism, right? If if you have, oh, I mean, let's just say humans, the rate is super slow, right? So if you take the entire alignment, assuming you never be able to do that, but if you chart the entire alignment beast, it's mostly going to be exactly the same size. So that might not be what you're targeting at all. So it depends on if you the best thing you can do is knowing where the SNPs actually lie. So for dengue virus, if your SNPs are in the E gene, that's super important to know. Or like if you have dengue virus and then somehow you have no E gene coverage, that's terrible because all the mutations happen in the E gene. So if you don't have any coverage of the E gene, you literally are missing so much evolutionary history of dengue virus. So it really depends on the data set. Just doing SNP studies, nothing wrong with that, but it's contextual. It's based on what you're actually trying to look at, what questions you want to answer, how the genomic structure, if there is genomic structure, how that is reflected in the SNPs, things like that. So yeah, you probably see with all my answers, it's never like a yes or no, or like you have to do this or you have to do that. Anyone who thinks that they can automate beasts I don't think is doing the right thing. But that's my very personal opinion.
Thanks.
Caitlin? No worries.
So kind of building off of Kelly's point, so when would this using like your SNP alignment as input for beast, would that be different from, say, if you made a tree based on your SNP alignment and then you use that tree as input for beast? Would that be or would you still have like the rate would still be like 10 times what you would actually see?
What do you mean by like using the tree as an input?
Like the Nexus file from the tree you've made?
Okay, to make it clear, the main input of like beast is the sequence alignment. So that's the data. The tree itself isn't data. The tree can be placed as a prior essentially. So you can be like, I think the tree looks like this, and then you can make it. You can either fix the topology, so you can fix the tree generated by beast, and what that means is all my tree topologies will look exactly like this. So if A branched off of B, and the parent of A and B is the most recent common ancestor of C, you can fix that and say that my tree always looks like this. But that's not data for beast. So beast still needs the sequence no matter what. I hope that's clarified. But I know that doesn't answer your initial question. Sorry, could you ask the question again?
Yeah, so thank you. That's super helpful. I think that does kind of answer my question, although I was wondering like if you have like a rough idea of how large a genome is to be too large to do like the full alignment in beasts. Say if we're only doing like less than 100 genomes just to
okay, less than 100 genomes. If it's less than 100 genomes, I think again it really depends on all your models because as I said earlier, the more complex your models are, the longer it takes to calculate. So if you're doing like a strict clock and HKY, you could probably get away with like 100,000, no, probably more like 200,000 base pair alignment with 200 plus sequences. And that should take hours, maximum maybe a day. But as you increase the complexity of the models, I really don't have a plot for this, but you guys can see my browser now.
Yeah.
Okay, so it'll take longer for this thing to reach this state. So it might get stuck in places. Like it might get stuck here. So what happens is what's actually going on here? Basically, in your MCMC, in your Markov chain Monte Carlo, between each of these steps, so like between 0 and 1,000, you're changing a parameter very slightly. So you could be changing how the tree topology looks. You could be changing your rate parameter. You could be changing any of the parameters. And so at each point you're changing parameters, it could take 10 days for you to reach this sort of what we call equilibrium, or sometimes like what we say convergence. So what happens here is like, okay, I think that all of my parameters have reached a reasonable estimate. And then it should stay like this. So the more complex your models are, the more free they are to change, the longer it takes to reach a point at which it becomes stable. So, with the simplest models, you could get away with a lot more sequences and a lot more data, a lot more sites. But if for me, like when I start using micromodulator models, I don't like I can't tell you now that it will finish because again I haven't applied the models yet. But because it's so free to change, like it could be jumping between these two models like infinitely, and it could never converge. I don't have an answer for that. So it depends. Like I know you don't like to hear this. I don't think anyone likes to hear this, but like, it depends.
So
Okay, thank you.
I have a proposition. It's not a question. I think that many people are interested to talk more about beast. I have the impression that everyone has specific question and stuff. So what do you think if we do like a SPAAMTisch session like we used to do rather than a seminar, like a clinic, a discussion on like just like this, just questions. It's Beast, so I don't know if John is interested. We do a session just free question open. I don't know. Well, who?
Yeah, sure. I'll just find a time when I'm free, and I'll be happy to attend.
On the floor, I mean, raise your hands if you are interested or something like that. Maybe the reaction things like. Okay. Yeah, okay. So, yeah, obviously, like, yeah, a small group of people, we can do like something like this, yeah, and take like a full hour of questions, free open question when John has time, and I guess also like drop him an email as well.
Yeah. If you don't have my email here, I will. I mean, I'll write it down here.
Yes, I should have CC'd you in the email I sent to the spam community. So I guess people should have your email, at least those who are in the list of the spam email. But I think, Bianca, you can stop the recording if you want. I'm not